All right, so I am going to be creating a video for you to watch so that you know exactly how to do the 2 techniques that you need to know for this lab, week 5 lab. The first one is subculturing, and the second one is an isolation of pure cultures. So the purpose of your work for this week is to show me that you can do these things. So of course this is an online class, and so you don't have access to the laboratory as we would in an on-campus class. So what we're doing is replicating that in our own homes. And we're doing that because it really is important for you to be able to do this hands-on and to kind of really feel what it is to do this. But I think the fun part is that you can use things that are household items that you have at your house in place of the actual laboratory items. So I'm doing this at my home. I have different things that I did bring from the laboratory, like the Bunsen burner, but I don't have everything, so we're going to, and I don't have gas to hook up the Bunsen burner, so we'll be improvising for some of those things. So the point here is to get creative, to get things that can take the place of these things, and I'll make some suggestions as we go. And then to make sure that you're doing the actual technique. So there are multiple videos on YouTube that you can watch in order to go through these techniques and make sure that you understand these techniques. Also, of course, I'll be going through that here with you today. So the first one is subculturing. So if you're in the actual laboratory, I'm wearing my lab coat. Something that you need to mention is the PPE. So what you'll be graded on is making sure that you do have the appropriate PPE. If you don't have a lab coat, you can put on some other flannel or a shirt or anything just to indicate that you understand that you're putting on a lab coat because we're in a laboratory. Second of all, you would want to wash your hands so we can wash our hands, and then we would put some gloves on. I'm going to pretend to put some gloves on and you can do the same. So we can put some gloves on to make sure that we have our PPE. So we would do that. We would also spray down the surface here. So I would take some spray from the laboratory, depending on the laboratory that you're in or you would be working in, would be different type of chemicals, but we would spray down the surface, wipe down the surface to make sure everything is clean before we begin our lab. So make sure that you notice those things or mention those things in your lab because you'll be graded on them. So what we have here, I have all of the supplies that are necessary for both the subculturing and the isolation of pure cultures. I'm going to start by gathering some things. We'll start with the subculturing, which is where I'm going to use this. We're going to pretend that this is actually a mixture of bacteria. I'm using all sterile items here today, so I won't be actually using the Bunsen burner to sterilize things as we move along. So I'll be doing that just like you should do that at your home. But I will actually be using these things, but they're all sterile, so don't worry, I'm not contaminating anything. So this will be our broth of mixed bacterial culture. You can just use whatever bacteria. I guess I'll pretend this is E. coli and Micrococcus luteans, or luteus. Then what we're going to do is subculture to this slant. So you can see that it is slanted. It has the agar that's inside of there, and it is in a slanted form. This one happens to be some blood agar, some chocolate agar, but you know, for our purposes, we're just pretending any sort of slant will do. So that, and then for transferring, I also have some other test tubes here. So I have another test tube broth here and a test tube broth here, just so that we can see the differences here. I also have an inoculating needle, well, multiple, or an inoculating loop. So I will show you that. This is the inoculating loop. And then I have inoculating needles. So you should know all of these different things. The inoculating loop, the inoculating needle, the difference between a broth and a slant. This was actually meant to be a deep, but it melted. It was a gelatin. So a deep would just be agar that was filled, and it's more solid. It wouldn't be liquid when I turned it. I also have some agar plates here. So these just have a typical nutrient broth on them. You can see inside here. So I have 2 of them. We'll only be needing to use 1. And then I also have a Sharpie marker. So in order to subculture, what we're going to be doing is we're going to be taking our bacteria. So we're going to say we're just moving bacteria from here. And we want to subculture, meaning move it to another culture or create a new culture on something else. And so that would be in our slant, for example. Also, oftentimes we would subculture into a broth, and that's so that we can get a fresh culture, because over time, our cultures will use up all of the nutrients inside of the broth, and then they will start to die off. And we don't want that to happen as we're using organisms in the laboratory. So it is important, and often in a laboratory, you do need to subculture so that you can maintain the health of the organisms. So we are going to go through the steps of doing this subculturing, and then we'll go through the other steps. So First, I'm going to take my Bunsen burner. In the laboratory, I would be attaching this to the gas. So attaching it to the gas, when I start, I want to make sure that these down here, I don't know if you can see the holes, are going to be covered up. And then I would attach this to my gas and I would slowly turn on my gas. In order to know whether your gas is turned on, you would want to listen so that you could hear the gas. And then you, of course, would back your face and head away. And then you would use the striker. I don't happen to have a striker, but you can manage, if you haven't used one before, it's like a match or something, to light the flame. So I'm going to have my pretend flame here. Oftentimes when you start using the striker, the flame will be a little bit unruly, and then you'll want to use this little guy down here to kind of adjust how much airflow is coming through. Adjusting the airflow is going to allow you to get a nice cone like we have here. So notice that this cone has yellow on the outside and that we have a cone of blue in the inside. And that's what you're looking for when you get a Bunsen burner going, is that you want to have that nice blue cone in the middle. The blue cone in the middle is going to be the hottest part, and that's actually where you're going to sterilize your materials. So you want to make sure you have that nice blue cone, and actually it's the very tip of the blue cone that we're going to be aiming for. So what we're going to do for our subculturing is we are going to get our inoculating loop, and the way that we're going to do this is we're going to pretend like we are really in the laboratory, and I'm going to be contaminating these items. So we're going to start by holding the inoculating loop here, and what we're going to do is we're going to be transferring the organisms from here into our slant. So the first thing we want to do is sterilize our inoculating loop. So as I mentioned, what you would do is you would go to the very tip of this blue flame, and you want to get your inoculating loop very, very red hot. So very hot, you go up and down, getting it very, very, very red hot. And then you would remove it and allow it to cool. So you want it to cool, so you should indicate in your video that you're letting this cool for about 20 to 30 seconds. But when you're cooling, you don't ever want to be flinging it around to kind of cool it down faster, because as you're doing that, what you're doing is you're allowing it to kind of mix with everything in the atmosphere or in the area, and then that could get contamination into your sample. So it's best to just kind of let it sit so you get minimum amount of contamination if possible. So I would sterilize my loop. Then what I'm going to do is use this hand that's holding the inoculating loop, and I'm going to remove my lid. Now what we need to do is we need to flame the opening of the test tube. So same thing, you kind of move back and forth through the flame, and it's going to make sure that we kill any organisms that maybe were on the edge of this that are not our actual organism of interest. So then I'm going to put my inoculating loop into the broth, and I'm actually going to kind of change my view here. So I'm putting it into the broth. I can kind of swirl it around a little bit, and then I will take it out. Before I do anything, I'm going to flame the opening again for contamination, and then I will put the lid back on and set this down. So now I have, if this were not sterile, I would have my organisms on my inoculating loop. And I don't know if you can see this, but it looks a little bit like a bubble blower. So we actually have a little bit of a shininess, a little bit of kind of, you know, the bubble blowers that you put in, you get that film on there. And that film is what you're interested in having. So then what you'll do, we're doing our subculturing, is you'll open what you're going to subculture into. In this case, we're going to do our nutrient slant, or in this case, our chocolate agar slant. Similarly, you'll take the lid off with your hand that's holding the inoculating loop. I'm going to flame the opening of this one as well. Then I'm going to take my inoculating loop, and I'm going to go into my test tube. And I don't know if you can see here, but there's some liquid that's at the bottom of my slant. And I'm going to put my loop just above that liquid. So I don't want to go into the liquid. I'm going to put my loop at the bottom of the slant, just above the liquid, and then I'm going to zigzag back and forth all the way up the liquid. Then I'm going to flame this again and put my cap back on. Now, when we are incubating organisms, some of them are going to be releasing gases. So it is important when you incubate anything in a test tube that you don't put the cap all the way on. What I suggest is putting the cap on tight enough to where if you lift it with the cap, it doesn't fall, but it should be very, very loose. It should not be tight. You want to have that space, otherwise you'll have exploding test tubes. So we can set that down, and then we want to make sure that we flame our loop. So we flame our loop to make sure that all the organisms are off of our loop now. Notice that when I was going between the different test tubes and things, I didn't set this down on the table because then it would be contaminated. I didn't set this down in my test tube rack even because then that could be contaminated as well. So once you have sterilized your loop, you want to make sure that you continue holding your loop. So that is subculturing. If we were going to subculture to a broth, which you don't have to complete in your lab when you do your video, you just need to subculture once to the slant to show me that you understand the zigzag motion and going up above the liquid and things like that. If we were subculturing to a broth, it would be even more simple in that we would grab our culture just like we did, sterilizing between, we would open up our broth, and then we basically just would simply stick this in, swirl it around a bit, make sure that we get that kind of bubble film off, come on out, flame again, and then close it up. And again, we wouldn't want to make this tight so that we allow room for that gas to escape. And then, of course, we sterilize our inoculating loop. All right, so now what we're going to do, I'm all sterile here, recall, we are going to move into our isolation of pure colonies. So I have my agar plate. What I would recommend, by the way, for maybe when you're doing your lab, for the inoculating loop, I would think a pencil or a pen would be, or even a marker, would be ideal to use in place of an inoculating loop, because then you'll be able to show me exactly where it is that you're going to be putting your bacteria when you're doing it on here, which is important because you need to do that. For test tubes, I feel like you can kind of use anything. Maybe you can use a piece of paper that you've rolled up into a tube and taped, just so that you can indicate showing me, you know, going in and flaming and that kind of thing. I suggest for a Bunsen burner, you can make a little pretend flame like I did. Maybe put on a test tube or on a toilet paper roll tube, for example, that might stand up pretty easily. And then again, for the test tubes, maybe for the slant, you can do a similar thing with the paper roll, for example, and then draw a slant on it if that's easier for you. For the plate, I would suggest something like maybe 2 pieces of paper, so one for the actual bottom and then one for the top, so that you can show that you understand how to keep the lid on and turn it upside down, which is required. And then a marker, you know, a Sharpie marker doesn't have to be a Sharpie. We use Sharpie in the laboratory because it does write and stay on these things, regardless of being in the incubator, so heat or moisture or anything like that. So, but of course, if you don't have a Sharpie, you don't have to have a Sharpie, just something that will write so that you can show me each of these steps. So for our isolation of pure cultures, as is mentioned in the lab write-up, you need to take your bottom of the agar plate, so it would be the agar side of it, the one that has a side that is going to have the growth on it, and on the underside you want to write on it to make it into 4 quadrants. So I'm going to make my 4 quadrants here. Doesn't have to be perfect. So my 4 quadrants, just like that. Then also in the lab, I should have mentioned this with the subculturing, I skipped this part, but you also need to label on your test tube, so you should do that in your video, and label on your agar plate the date, your name or initials is what I suggest, and the organisms. And that's important because in a laboratory, you want to make sure if anything were to happen, if there was some sort of spill or something that happened and the organisms all ended up in this poopy mess somewhere, that you would be able to identify what they are in a laboratory so that you could indicate what level of seriousness that needs to be taken as far as cleaning it up. So it's always important that you label the date so we know how old the organism is, but then also the organisms. And of course your name would be so that you would know which one is yours in the laboratory. So I'm going to skip spending the time, but you would write that at the bottom here. So when I say the bottom edge, I don't mean the edge that's on the side here. What I mean is you want to write it along the curve here. So you would write your initials, you would write the date, and then your organisms along the curve here, allowing yourself the space to really kind of see what's going on with your organisms. All right, so now we are going to, again, take our test tube here with the organisms. I always kind of loosen it up before I do anything. That way I'm not struggling when I have my inoculating loop sterilized. So we are going to transfer this now pretend mixture of 2 bacteria, and what we want to do is we want to isolate cultures. And what that means is we want to separate the 2 of them. And we can do that using this technique. So when you are doing this, this is why I recommend a pencil or something like that for your inoculating loop, is so that you can show me that when you open this up and you go into your quadrants, that you are doing a zigzag motion as is written up in the laboratory, and I'll be going through. One thing that I didn't do while I did this, because I was kind of skipping ahead, was I didn't label my quadrants. So I'll label them 1, 2, 3, and 4. And that is just simply so that as I am taking this and I flip it around, I remember which quadrant is which, because sometimes that can be a little bit confusing. And then I don't have to worry about trying to remember which one's which from down and from up. All right, so I'm going to take my inoculating loop. I'm going to flame my inoculating loop to sterilize it. I'm going to get my mixed broth culture. I'm going to still be holding my inoculating loop. Remember to mention in your video that you're allowing this to cool for about 30 seconds. Okay. Opening this up, I'm going to flame the opening. I'm going to go in with my needle or with my loop, swirl it around a little bit, make sure that I get a good mixture of organisms, take it out right in the center as easy as I can. Flame that again. Put this back on. And now remember, I have my organisms here, so I'm not going to be setting this down anywhere. I'm not going to be moving anything. What I am going to do is I'm going to take my, I don't know if you noticed here, but I have my agar plate was upside down on the desk. So I'm going to lift it up, and now I have my agar. I'm going to go to my quadrant number 1. And in the lab, you can see that it shows how you should do your zigzag motion. So I'm going to start on the edge. If you can see this, start on the edge. And I'm going to kind of go the whole length of the zigzag or the quadrant and not get too close to the middle though. Leave a good, couple centimeters or so distance in the middle. We don't want overlapping in there. So now that I've done that, I would close this up in the laboratory. I would flame. So what I'm going to do is I'm going to flame this to completely sterilize the loop again. Now I'm going to let it sit for about 30-40 seconds to allow it to cool. Then I'm going to pick this back up. I'm not getting more organism. Now I'm going to go into quadrant 2, but I'm going to start in quadrant 1 and I'm going to pull organisms from quadrant 1 into quadrant 2. So I'm going to grab from quadrant 1 and then I'm going to go back into quadrant 1 twice. And then I'm going to zigzag a little bit more in my second quadrant. Of course, while you're doing this in regular lab, you wouldn't be talking right into it. So just, this is for my purposes, don't talk directly into your agar. So then I would sterilize this. So sterilizing my loop. Again, I have a sterile loop. Again, I'm going to open this up. Now I'm moving the bacterium from quadrant 2 to quadrant 3. So I'm going to start with my loop in quadrant 2, drag into quadrant 3, go back into quadrant 2, drag into quadrant 3, and then do a couple more zigzag towards the center. Not right into the center though. Then I'm going to flame my loop again, sterilize it. Now I'm going to go into quadrant 4. So I turn it again, grab, starting in quadrant 3, and zigzag, go back to quadrant 3, and zigzag, and again, relatively close to the middle, but not all the way. Close that up, and flame my loop. Now I let it cool for 45 seconds again, or 30 to 40 seconds or so, and then I set it down because that's sterile, and I'm not going to be using it. So now I have completed my 4-way streak. And what this does is, as you can probably imagine, going from quadrant 1, where we took the full strength mixture of bacteria, we spread that around, and then what we did was we sterilized the loop, which allowed us to pull just a little bit from quadrant 1 into quadrant 2. And then the same thing, pulling a little bit from quadrant 2 into quadrant 3, and then the same to 4. What that should give us is that at the end, you should get in quadrant 4 very separate individual bacteria cells. And then as they are being incubated, then those will grow into colonies that are separate from each other. In the laboratory, if we were in the classroom, we would have 2 different organisms that look different when they're growing. And so then you could very clearly see whether or not you succeeded at separating one organism from the other. So now in the lab, you want to make sure that you indicate that you are going to be taking your slant that you did your subculturing into, and that you would be taking your agar plate, and that you are going to put both of these in an incubator at 37 degrees C for 24 to 48 hours. One thing that you want to make sure in your video that you do is make sure that you show me your zigzags. So that's again why I mentioned using something like that. You should have your zigzags, you should have a cover so that you can show that you're using the bottom and that you're doing your zigzags and that you're putting it on the cover. Also, I want you to indicate and mention in the lab, so that I know that you know it, that when you put something in the incubator, when you put an agar plate in the incubator, that you absolutely have to put that lid down. So you can see here I have the agar on the top, and that this is lid down, and that's how I would place it in the incubator. And that's very important because when they're in the incubator, there will be condensation. And that condensation will accumulate, but if it's upside down, so lid down, that will accumulate on the lid rather than on your actual organisms and smear all your organisms together. So anything in an incubator on an agar plate needs to be top down. And here, remember that you should be mentioning that this is not all the way tight, so you can allow the gases to escape. I'm going to do the right thing, and I'm going to turn off my gas here, make sure that my flame goes out. And then what I would also do is I would spray the counter here after I put these organisms in the incubator, of course, spray my counter to make sure that it is sterile again. I would remove my gloves appropriately, and then I would wash my hands. So that is the lab. You don't have to talk the whole time like I did as I was explaining things, but what you do need to say and what you do need to talk about while you're going through is what you're using for what. So when you pick something up, if it's 2 pieces of paper, you will need to say, this is the bottom that has the agar, this is the top, the lid, when you're doing it. Or you'd have to say, this toilet paper tube over here is my Bunsen burner. And you need to describe what you're doing. So, not as in-depth as I did, of course, but you would need to say something like, I'm taking my inoculating loop, and say you're holding a pencil, and I am putting it through the top part of the blue cone, and I'm sterilizing it. I'm letting it sit for 30 to 40 seconds to cool. Then I am opening up my test tube. I am flaming the opening of the test tube. I'm putting my inoculating loop into the broth. I'm swirling it slightly, ensuring that I have a film in the inoculating loop, flaming the opening again, putting the lid back on, etc. So you need to narrate everything that you're doing so that we know that you know what you're talking about, and so we know what you're using for what. Okay? So then at the end, make sure that you lift up your agar plate and show me your zigzags. You can't see my zigzags here probably. You can see kind of in the agar that I did zigzag in. However, you can see in the lab exactly how to do your zigzags. All right, so if you have any questions, go ahead and let me know. Otherwise, make sure that you have one video that you've created. If you do it multiple times and you can piece them all together with editing, that's fine too. Just make sure that your submission is one video, as everybody will need to be peer reviewing, and it needs to be one video so they can see it. And also remember that it needs to be 10 minutes or less. Okay? All right, thank you.